Welcome back, students. So yesterday we discussed about one of the genomic marker or polymorphic marker technique that can be used to analyze different polymorphism among population. The technique is called restriction fragment length polymorphism, RFLP. We've spoken about different steps involved there, per se, 5 steps or 6 steps. The first step is common, digestion of the given genomic DNA, then electrophoresis to get a DNA profiling. Then of the common DNA profiling or random fragments here, you specifically probe your own RFLP based on the kind of DNA probes you already designed. So how do you do the probing? Probing you will do through hybridization mechanism. So you have the developed probes, that is nothing but DNA sequences. Then you first transfer the random fragments from the gel, from the genomic DNA to a nitrocellulose membrane. The procedure is called blotting technique. Here you are using a northern blot, and then go into that blotting technique with the hybridization with the probe. You at the end you get an X-ray film. Sorry, southern blot. So you get into a X-ray film that specifically picks up your probe in terms of the blotting of the given restriction digest of the genome. That is the fundamental way, and specifically you use restriction fragments because the restriction fragments have capacity to digest the DNA given the sequence specificity of a genome. A non-specific restriction digest, you may have different variable restriction enzymes on the given sequence. The straight line is the genomic sequence here, the random sequences. Given the process of restriction digestion, the chemical process and incubation, then you cut the genome into randomly at different places. But if you can already know the restriction endonuclease in a given location of the genome and two people, for example, polymorphisms here, human 1, human 2, genome is almost identical. But if the restriction sites given in the location are constant, then you can see that a specific size of the fragments can be obtained. These fragments can be analyzed in the agarose gel. So basically talking in terms of the fundamental or molecular basis of RFLP, it is nothing but it changes in a given DNA sequence, it changes in terms of variations or mutations. The mutation can come either by a specific terminology we use in terms of point mutation, that's substitution, base substitution, or insertion and deletions, that is also called. One more thing, through transposons, rearrangements of the genomes can happen. So if these variations happen in the given DNA sequences among the population, then the variations can also happen in the restriction sites of the given location. So using these variations, the restriction sites are also vary. They can change a location given a sequence, causing either gain, loss, or relocation of a restriction site among a common population, among humans per se. What I am talking, if you look in terms of the fundamentals of it, the molecular basis, you take case by case study here. This is nothing but genome variations among populations. How the process happens? Through mutation and polymorphism. One case is that insertion causes increase in fragment size. So you have an initial sequence here. Along with the genome sequence, you have two restriction sites that are common per se. A change happens in the given genome population, that new insertion of a new sequence happen during evolution or during the course of variations. So during that, so if you digest the fragment before the change, you may get a specific length between them here. And then if you digest the same using the same restriction enzyme, digest the genome, you might get a different size here because it's a variable size due to an insertion. The same thing you're talking in terms of a human 1, human 2. Though I myself and yourself, we may have the same restriction site in the genome, I might have a polymorphism site, I might have an indel in my genome in the same location, causing that the size may vary. So restriction fragment size varies. That is the fundamental basis of RFLP to distinguish human 1 to human 2. In another case, it may happen deletion, the reverse. Already we have it, fragments, and already a variation is here. Due to some change, this portion might be deleted. So we are going in a reverse direction from longer fragment to the shorter fragment, keeping the restriction site as a constant. This is another case, deletion. Insertion here, deletion here, combinedly they are called indels in a given genome of variation. Now you take a case of substitution per se. You have the same sites. In all the cases, we might keep the upper strand common. Same sites are there. Suddenly, a substitution happens, a mutation happens in a given population. A site, instead of G, an A can be incorporated in the changed sequence. This A, probably a part of the restriction site, a sequence change happens here, so that this modification can duplicate a restriction site here, given a 4 base pair, 6 base pair cutter. Then you are adding an additional restriction site here into the new genome. By doing so, if you do a cut, instead of cutting one single fragment, this altered genome sequence will give you two fragments, one small and one long. So it is also one more fundamental change. Causing this change, new substitution can give a new restriction site. The opposite that, it may delete the restriction site per se. A change happened here in the middle. Per se, a change happened in one of these end of the nucleotide. The length is same. Sites are here in the normal. Mutation happened. G has been changed to A. Given that scenario, the mutation or restriction site has been eliminated. If this site has been eliminated, basically you may get one fragment here. In this case, you may not get a fragment at all, because if you get a single cut of the genome, it will become linear. It may not, it may give linear, and it may be give a size control to the genomic DNA itself, but it may not give a specific band you are looking here, a small band fragment you are looking. So elimination causes disappearance of the restriction site. And the rearrangement, an example of rearrangement, you have several transposon or repetitive elements in the genome sequence. You have two restriction sites here. Coming to human 1, human 2, a transposase function happened, retrotransposons. They moved this site into another location in the genome, given the specific length here. By moving so, a restriction digestion can give variable size of the DNA fragment. So thus, causing these variations, causing these variable forms, this is the fundamental basis of polymorphism or haplotypes given human populations. How you can use the restriction digestion sites in a given location to identify different polymorphisms solely based on the size of the fragment that you can use. That is nothing but reflect. So one more important concept in terms of the molecular markers we are speaking, we are talking a general concept of a marker is dominant or it's non-dominant. You say one thing, dominant, non-dominant, or co-dominant. You say two concepts here. In another concept for the marker, you are talking a single locus marker or multi-locus marker. So these two are really fundamentally important. You are saying as a dominant marker, if a marker is dominant per se, it can identify only one allele. It can give only one band. Either the band is there, allele disappeared, or the specific genome fragment disappeared due to mutation. Then it will tell the dominant marker that one band will show if the marker is there. If it doesn't, it's eliminated, absence of a band in a dominant marker. That is the only case. But in coming on to the point of co-dominant marker, it gives, in a normal condition only, it gives two bands per se. And then if one band disappears, at least one band stays. So co-dominance is always unique in terms of saying that both alleles are important. If one allele is mutated, but the other allele band can appear. Per se, you look in a situation here, homozygous situation dominant, you get only one band for AA. And then BB is kind of co-dominant, it already has two sites. And then given a heterozygote here, AB, it's a co-dominant phenomena. That's why you get one band from here, from A allele, two bands from B allele, you get three bands in the AB allele. This is in a situation of co-dominant marker. If let's say the marker in the given location is a dominant, in any given condition, you may get only one single band, that 111 here. So either A present, AB present, you get one band of the common size. But if BB present, you may not see the band at all if a dominant marker. In a recessive marker, you have to count on whole in terms of heterozygosity bands, homozygous allele, depending on the restrictions. That is really important. And in consists of multi-locus marker, you are saying that in a given genome, the marker not only looks in the 8 kb fragment, it can go to 10 to 20,000 kilobases size. it can cover that much of the length of the genome. By covering so, it might cover more than one gene, more than one locus. So that is called, if a marker covers a bigger portion of the genome for polymorphism, it is called multi-locus marker. But if it covers only one small portion per se, single nucleotide polymorphism, SNPs, then it is a kind of single locus marker. That is very important. So thus summary RFLP reflect in if two organisms differ in the distance between the sites of cleavage of a particular restriction endonuclease, the length of the fragments produced after digestion will differ, and this is the fundamental, the difference and similarity of the bands. You can deduce the allelic composition or allelic mutations or variations in the polymorph. That is one important. In summary, reflect uses one of these concepts. It's a codominant marker, so it depicts both alleles, so both bands will be there in the gel. It is a non-PCR based method, so no primers are required. And starting point, you need a large amount of purified DNA in micrograms, tons and tons of DNA, genomic DNA you need to isolate. And pros is highly reproducible, but the cons is that it's very time consuming, very laborious. It's not easy to do molecular biology in the lab given a condition. You need to run a lot of gels. Sometimes it doesn't work. You need to repeat. So coming on to fundamentals, digestion with restriction enzyme, separation, transfer, hybridization, and visualization. One more con is that it can detect only insertions and deletions of large sizes. This polymorphism, this marker technique, because due to gain and loss of these sites, but it is unable to detect small variations in terms of the point mutations or a little bit small variations. It is unable to detect that. That is one of the disadvantages. So I have given you a little bit more lecture with descriptive notes. You can read it and make notes of it. Non-PCR, codominant. RFLP can cover 1 to 10 loci detected. So that's why it's called multi-locus marker. You make notes of it, advantages, disadvantages, and then little bit pros and cons and applications of it. Any marker, the application is DNA profiling or fingerprinting, criminal cases, disease status of an individual, and also in terms of polymorphism among human populations, you can identify that. Look into that and make notes of it. And thus summarizing the reflect, I will make another video of some of the thing we missed yesterday. I will make 20 minutes video and upload that. Coming on to now, so this is a non-PCR, basically restriction digestion based method, and mixing with Southern blotting and visualization of DNA probing. But given in the last class, we discussed different markers, PCR, non-PCR and PCR based markers. So you see the fundamental difference here. Wherever A comes in a given marker, RAPD, AFLP, that is nothing but you are amplifying the source from the given genome. In here, you are cutting it, but in this case, either you cut or you amplify starting from the material. You will always use a PCR. By default, if you use a PCR, then you are using a primer to amplify the given sequence of interest. So this method, two methods are PCR based. One is AFLP, one is RAPD. So AFLP again, common to RFLP, there is the same alphabets, FLP, FLP, that nothing but fragment length polymorphism. These two techniques again commonly use restriction digestion is one, and this method doesn't use PCR. This method uses PCR based on the restriction enzyme sizes. We will see that what is the advantage it uses. Then RAPD doesn't use the restriction enzyme. It directly from the genome randomly amplifies. That's saying that this marker is codominant marker. These two markers are dominant markers. You may not find two bands given the alleles. You always find only one band in this technique. And then as you suggested, no primer here, PCR is required, these two. In this case, AFLP, you will have a selective primer. You already know the primer, you will design yourself. Why I'm saying selective? Because you use the restriction enzyme digestion sequences. I will see that and I will show in further slides. The restriction digestion portion are given a location of the genome, how it cuts, what are the sequences, unique sequences of restriction enzyme, what are the sites. Based on that, you can design a specific primer to amplify a fragment portion of the genome from the restriction enzyme 1 to 2. That's why you call selective primer. In RAPD, it's called random amplification. RFLP is a amplified fragment length polymorphism, random amplification polymerization. We will see that here, RAPD. So in terms of highly reproducible, row reproducible, you need small DNA quantity, you need large quantity, and also very purified quantity here. We will see once we read it these two more further, you can come back and analyze this technique again one more time, make notes of it. This table is very important. If you understand the differences in each sentence here we are talking, then these three concepts are very easy and very important in terms of exam point of view. So AFLP is nothing but amplified fragment length polymorphism. FLP is common, like you can say it's A-flip or reflip, F-flip, I don't know. So amplified, you are amplifying the fragment length from the genome and the restriction dilatation. Basically, this method was first introduced by Vaughan and Jabo in 1993. Again, whatever the year we are talking, that year brings an hallmark of new molecular biology technique. So PCR might have come up so far. So that's why they could able to use PCR as an advantage of polymorphism or markers here. It involves a combination of reflip and PCR, saying that the reflip, it uses the restriction digestion process again. And further step, it uses PCR technique. And it amplifies the same gene from different individual to find out the variations or polymorphism. It's a very powerful technique to detect the polymorphism. So why I'm talking PCR here, you need to again remember the fundamental difference. In reflip, after restriction digestion, you use a specific probe, DNA probe, to hybridize to your restriction fragment you are interested. That is a reflip you are interested. So you do hybridization, DNA hybridization in that technique. But here, you don't do the DNA hybridization, you don't make a probe per se, but you use a specific PCR mechanism. That per se, what I'm saying, in terms of a normal gene here, in a normal condition, and a disease condition here, you are seeing three restriction sites here in the normal condition of the gene. That restriction enzyme is MST1. But somehow, due to some mutation, there is a disease occurrence in the populations. When you look on the fundamentally the molecular mechanism of how diseases happened at the gene level, they were able to identify that a restriction site has been eliminated in the mutated version or disease condition. So per se, in this scenario, if you make a probe in terms of the restriction digestion and in terms of the southern blot, you may not get three bands per se in the disease condition. So you get the bands here, three restriction site, band 1, band 2, you may get probing to this region here. You make the probe common to here, you may get a normal condition 2 bands, but coming on to the disease condition, you may get only 1 band because this site has been eliminated. Then if you do the digestion, you will get only 1 fragment here that is larger than the fragment here. So you see the 2 bands in the normal condition. In disease condition, the fragment is size is higher. So it moved up in the agarose gel, smaller fragments move fast. So basically by developing the probing also, you can see that coming to the normal disease condition, this is a codominant markers again. In humans, it's always codominance. In normal, you should see 2 bands given the sites, but in disease condition, you see 1 band. So this is, you are using probes here in a specific location. But in AFLP, you move too much of PCR method. You design a specific primers in the given location here. You design the primers will amplify. Again, the primers may amplify only 8 kb here only, but you will use another method called a restriction fragment specific primers. You use an adapters to identify each restriction site. Based on that, you can identify only this much fragment. You can identify this whole fragment in the given disease condition. How does it happen? So in AFLP technique, it is a technique based on selective principle of selectively amplifying the subset of restriction fragments from a complex mixture of genomic DNA, genomic digestion, and then by restriction endonucleases. So basically, the first step is common to reflip. You take the genomic DNA, you randomly digest it, but you pick a specific endonuclease, you digest it. From that, you try to amplify the given genomic sequence to see that what are the changes in the genome happened in the vicinity of the restriction enzyme site or flanking regions. What I am saying in terms, so basically the following steps will be there. The digestion step is common, and the third step is amplification. That means PCR is bringing. It may be hybridization in reflux. Here you are using PCR, and electrophoresis is common. But most important step here is you are using another step called adapter ligation. Between the digestion or amplifying specific restriction digestion product, you use a specific method called adapter ligation. I will explain that. What is it? Why we are using it? Because when you want to amplify a specific digest product, you are using PCR means you have to design a primer, and the primer should be specific to the restriction enzyme you are looking for. You don't want to randomly amplify primer along the genome wherever you want. You may not be getting any specificity, but you are already digesting with several restriction enzymes. You have your own restriction enzyme fragment. Then why don't you design a primer of specific to the restriction enzyme and amplify only that much region, given the usually PCR can amplify 1,000 base pair regions, good enough, but 5 to 10,000 also it can amplify, but 1,000 to 2,000 is a good start for accuracy. So how do you do that? So you need to better understand first how restriction digestion happens in a given genome, what are the sites. At the same time, you can ligate a specific adapter to the restriction enzyme and the 5' end. We will see that. So first, fundamentally understand a little bit on restriction digestion. Basically, how it happens. Restriction enzymes are restriction endonucleases or exonucleases. They are the proteins that cleave DNA at a specific location, right? They are the scissors. Double stranded DNA will be there. They can cleave based on the specific complementarity sequence. Remember that restriction enzyme, it will cut only a double stranded molecule. It will never cut a single stranded molecule given only a sequence of one strand of the restriction site or restriction enzyme recognition sequence. It needs a double stranded having both sequence complementarity. Then only it will cut it, and then it is coming from bacteria, and then they are indispensable tools for molecular biology and biotechnology in terms of cloning. They are endonucleases if they cut within the given sequence, or internal cuts. Exonucleases, they cut in the outside, and they may be a 4 base pair cutters, 6 base pair, and 8 base pair cutters, different variable enzymes. I think so far we have almost 250 or 25,000 enzymes with having specificity of 2,500 enzymes, specificity of 250 enzymes have specific sequence identities in a general terms speaking. But given given a population of the genome in terms of the genome base pairs, human genome is 3.2 billion base pairs. How many restriction sites or restriction enzymes you can find among that humans? So what frequency you can find? Basically, given the 4 base pair cutters, the frequency is 4^4. In a given 256 base pairs multiplication or of the distance in the genome, you can find one 4 cutter, and then given a 4,000 base pairs, you may find one 6 base cutter. One 8 base cutters are very rare. You might go around 65,000 base pairs length to find one cutter of 8 base. So by basically saying is that in a given genome, a random genome we are speaking here, there will be a different restriction sites. These whatever I'm saying, these black bars, these are nothing but restriction enzymes. You are looking at the genome from 0 to 35 kilobases, basically speaking, the length of the genome starting from 0, first nucleotide, 35,000 base pairs, kilobases. Then given this restriction enzyme, how many sites are there in this length? You can find there are many kind of average sites here. And given AGA1, there are more sites saying that it may be a 4 base pair cutter. The less frequency, it is 8 base pair cutter. Like that, in a given same sequence, multiple restriction digestion restriction enzyme sites are conserved. No one matches with the other, right? We are moving along. Each site is unique given the location, given the restriction enzyme. But in a given distance, you may find multiple restriction enzyme site, multiple sequences of that. So per se, if you cut one genome here with the restriction site, if then you cut it, then there may be the two portions of this may be cut. One single cut, let's say, I divided this genome into 2 fragments. Then restriction digestion, I may follow ligation again, right? I might use a ligation reaction to stick it back. I cut it, I want to ligate it back. So I can use ligase enzyme to stitch the given same genome sequences. I can do that. But you remember that when you cut a restriction site, the flanking sequences 5 prime 3 prime, you will have a genomic sequences that flanking the restriction site. So the restriction site sequence is nothing but it's a genome sequence itself, having a unique identity for a specific enzyme. It is not coming out of the genome. It's part of the genome. So ATGCs are common. Given the ATGC, one sequence specific may be to one enzyme, but it is flanked by all the genome. The enzymes may be part of the gene, non-coding gene also. So you should always remember that these are not added artificially. These are internal part of the genome itself. If you cut it, you may have flanking sequences that correspond to the genome itself. If it's a gene or non-coding, it corresponds to that only. So you need to remember that fundamental difference. And also, when you do a restriction digestion, specific to the restriction enzyme, you may get sticky ends, you may get blunt ends. What I'm saying here, sticky ends are nothing but 5 prime protruding ends. You cut this specific Unit3 digest, it will give 2 hanging regions or sticky ends, protruding ends. They are 5 prime region only protruding, 3 prime has been cut. And the same way you use PST1, 3 prime is hanging, 5 prime is not hanging, so protruding. So one enzyme can give 5 prime protruding ends, one other enzyme can give 3 prime protruding end. And then in another case, there are no ends, the blunt end will be there. So for example, if I take these blunt ends, I do the ligation reaction, I may not be successful, because if you want to ligate these cut restriction enzyme cut sites back again, I can give nucleotides specifically here, TCAGA, complementary nucleotides, and I can start ligation reaction, 5 prime to 3 prime, A will come, G will come, C will come, T will come. I can stitch the site back based on the protruding end. This sequence can use it as a primer, or complementarity can be used. But what about blunt end? I want to ligate this back. I cannot do it. There is no stitching back, no 3 prime OH is free. But what nucleotide on the down in the 5 prime, I don't know. I cannot match it. So to do the blunt end ligation, what we will do, so we know the restriction site sequence here, GAT is here, the cut ATC will come. If I can add TAG here, for example, from this direction or 5 prime 3 prime, I add specific complementarity sequences here, then I can make them as a protruding ends. By doing so, I can stitch it back the blunt end ligation. So that is fundamental technique. This is really important for AFLP. How do you design a primers for a given restriction site? And remember that in a given cut, there may be after this restriction site, protruding or flanking sequence this side or that side is genomic sequence. So if you stitch it back, you are bringing it back the genomic sequences outside of the 5 prime of the restriction site. So this is important. Restriction digestion, ligation, blunt end ligation, you need to add specific nucleotides given the complementarity of the enzyme. Then you ligate it back, you stitch it back. So how we can use this technique in terms of AFLP? AFLP is nothing but you have a restriction site 1, restriction site 2. The one is EcoR1, the second one is MSC1. This is the restriction site sequences in the given genome. The other genomic regions are here. You perform the restriction digestion, you may get 5 prime, 3 prime overhangs as suggested here. And in between, you have the genomic sequence. Keep remembering that. And then you might want to recognize this portion of the restriction digest, whatever is cut here, whatever cut here. Basically, this cut portion is nothing but this is one half, this is the other half. Given the restriction sequence, restriction enzyme sequence in 5 prime, 3 prime, given the different halves of it, you have the control over the sequences. Then if you are stitching back, I know which portion of it I have to bring it back to make the site to bring the site back in the genome. To make from here cut to ligate, I can bring an adapter sequence that specifically brings the complementarity sequences here. So you develop a specific adapters called adapter molecules in terms of the DNA nucleotides. You design them based on the restriction site or hanging sites coming from. Then you ligate them back. You ligate them back into the restriction digested fragment. So given the whole genome, if you undergo this digestion, this is the fragment you get from the gel, one size. You isolate the DNA, purify it, then you ligate it with specific adapters. You make it again blunt, leaving the other genomic sequences. From there, what you will do, nothing but adapter 1, adapter 2, bringing a specific enzyme ligase. Then you stitch this region back, ligation. Then if you go and change in terms of the ligation and given the nucleotide sequence again, you might have to work out, you might have to take a pen and paper, you might have to work with different enzymes. What is the sequence that after cut? Then you need to recognize different kind of internal sequence from the genome. And if you make an adapter, this is the black. In a given sequence here, 3 examples. The core sequence, the red ones are after restriction digestion. This is the core sequence or the protruding or blunt end coming after digestion of EcoR1, PST1, MSC1. And then from that you go upstream, you randomly select some nucleotides and different specific nucleotides. You design a specific adapter DNA molecule, you stitch them back to the site. These are the adapters specific for a specific restriction digestion. Then coming on to the internal sequence here, for example, this may be G, may be the protruding end, given that you stitch it back with a C, coming that this is part of the restriction digestion internal sequence that you stitch it back with complementarity. So you basically you develop a technique in terms of adapter ligation. Given the restriction digest, you attach additional sequences, you try to deduce the sequence and recapitulate the genomic sequence there. The green is internal part of the genome, internal part of the restriction enzyme site. The red is the core sequence, and this adapter sequence you are added further. This is really important. If you understand adapter ligation better, you will understand AFLP easily. So once you attach that adapters, then you need to identify the polymorphism in a given sequence. You move up in down this downstream region. You can get sequences from human 1, human 2, amplify these specific location in between only, and then compare these sequences that any changes are there, deletions or insertions are there. Based on the size you get from the restriction digest, you can identify. So how do you do that? You don't need to simply go again restriction digest. You need to amplify, picking a specific restriction site here, amplify the given genomic location internal to that. Then to do that, you need to PCR. To do the PCR, you have to design primers again. So now this is your DNA 5 prime 3 prime 5 prime 3 prime. From this sequence, again you have to develop primers, taking that this adapter sequence you already know. From this, you can develop some primer, homologous or complementarity regions. From this, try to expand this internal sequence, this direction, you get a PCR product, you analyze the PCR product. So how do you do the PCR? It's nothing but given a double stranded DNA in the genome. In this case, double stranded digest in terms of coming from specific restriction digest. You know some of the sequences already here up to the point of restriction enzyme. Then you amplify with specific primers you design. Then you get a lot of multiplication of the DNA or interest of the genomic location. After that, you can analyze them. So how do you design the primers? After ligation of the DNA with adapters, the first thing you will do that, you do a pre-amplification step. It's nothing but given the cuts here, given the ligation here, you use the downstream and complementarity when you design the primers. You need to remember that if you design a primers, you are using a sense strand, anti-sense strand. You have to be very careful in that. We are not designing a primer to this strand. We are designing a primer to the downward strand. The first complementarity comes in the cut region is the T here. So A will be added here. And then in the other region is called, you use this opposite G. You have to add a specific primer with the C. Why I am saying is that it is internal to the restriction site. You need to amplify that. If you don't amplify this properly, you simply add a primer to this sequence and amplify. You are missing a C here, you are missing a here. Then basically you yourself are introducing a kind of specific mutation in the PCR amplification. You don't want to do that. You need to regain the restriction sites when you amplify, then the whole sequence will be stitched back in a normal way. So once you do pre-amplification, then you do other nucleotides you add, then you amplify further. It's nothing but while you are adding the primers, you are adding extension sequences, the blue ones here. You may add one, you may add no extension sequence given that you rescued the site normally, or you can add one nucleotide, you can add two nucleotides. This is called extension nucleotides for the primer. That is called MSC primer plus 0, primer plus 1, primer plus 2. So once you need to remember the adapter ligation, you need to remember the primer, how you selected the primer, how do you replace the nucleotide sequence, how do you rescue the restriction site, and then added a specific primer. Then you simply amplify and get the different sizes and then look into the difference. So this is a text, explains everything I explained, primer in different colors, enzyme site, core, restriction remnant site that you rescued. You have to go and look into that. And I have added additional material that beautifully describes most of the process. One more cartoon. You have to make notes of it, read into that one more time. And conclusively, the AFLP is nothing but total genomic restriction digestion, adapter ligation, pre-amplification, selective amplification, and run the gel and look into that, and then advantages, disadvantages. So tomorrow we will take RAPD. Okay. We will again do a lot of revision classes. We take everything and revise one more time. Okay. Thank you. See you tomorrow for RAPD.
Thank you, sir.